Hello, and welcome back to Mammalian Anatomy at Keuka College. Today we're going to be starting our lecture series on Chapter 16, the Special Senses. And in this video, we're going to be focusing on taste, or if we're using the physiological term, we call it gustation. All right, let's get started. So before we get into taste specifically, I do want to talk about how both taste and smell are chemical senses. These senses actually rely on chemoreceptors, so chemical receptors, that will actually bind to either food or odor chemicals that are within our environment. So for taste, what really happens is food chemicals are dissolved in our saliva, and those food chemicals are called tastins. And these tastins will bind to chemical or chemoreceptors located on our tongue and other regions of our mouth. And that binding will stimulate the taste receptor, and that taste receptor will activate a sensory neuron as a response. Smell is also a chemoreceptor kind of based system. Here what will happen is airborne odors will dissolve in the mucus of our nose and bind to chemoreceptors in our nose. And again, that will activate sensory neurons, and those sensory neurons will help carry that information to the central nervous system. Taste and smell are very, very closely linked together. So not only are we going to talk about them based on the fact that they are both chemoreceptors, but they do work together to allow for us to be able to taste many, many different flavors in our like when we eat. So let's get into talking about taste in a little bit more detail. So first and foremost, I wanted to talk about taste buds. Taste buds are essentially going to be the location where the chemoreceptors are going to be found, and they're going to be the specialized organ that is going to facilitate tasting or gustation. And taste buds can actually be found in many different areas. The pharynx, which is the back of the throat, the inner lining of the cheek, the palate, which is the roof of our mouth, and even the epiglottis. The epiglottis is an odd structure that is in our throat. It actually functions to close our airway as we're swallowing food, so food doesn't get stuck in our larynx and our trachea. But there can be taste buds located on our epiglottis as well. But the majority of our taste buds are going to be actually found on our tongue. But notice that it's not only going to be on the tongue, there are other areas that will have taste buds as well. There are three major types of taste buds that occur on our tongue. There are going to be the, sorry, my apologies. Taste buds are going to occur in areas called papillae on our tongue. And there are going to be three major types of papillae on our tongue. There's going to be, first and foremost, the fungiform papillae. This is going to be the most predominant type and is going to cover most of the surface of our tongue. Over here you can see that they're the small little dots. They give our tongue a little bit of a rough texture to it. On the sides of our tongue, we're going to have these kind of slit-like papillae. These are going to be the foliate papillae. I think of like foliage or leaves. They almost look like leaves. Fungiform refers to kind of having a fungus kind of structure to it, so that's a way to remember. And then we have the valate papillae. Valate papillae are going to have a V shape to it, so that's how it gets its name. Just be careful when you are looking at the anatomy of the tongue that you don't mistaken these structures for taste buds or papillae. What these are going to be our lingual tonsils. So here are our palatine tonsils, and these are the lingual tonsils that are going to be attached to the tongue. So those are not papillae. They are going to be actually a part of our immune system that provides kind of protection as food is entering our mouth and our digestive system. Over here, what we've taken is we've taken a valate papillae and kind of zoomed in a bit. And you can see here the taste buds are actually buried deep into the papillae and are located here in blue. This yellow color over here is actually going to be the sensory neuron that is coming off of the taste bud. So let's look at the taste buds in a little bit more detail and kind of look at its structure. So we are going to be looking at one of these little blue structures over here, and we've zoomed in over here. Taste buds are composed of two types of cells. There's going to be a receptor cell and a supporting cell. The receptor cells are going to be these gustatory epithelial cells here and here. And these gustatory epithelial cells are going to be what are going to be responsible for housing the chemoreceptors that are going to bind to the taste ends or the food chemicals that we are trying to taste. Those chemoreceptors are actually going to be on the gustatory hairs. There are little hair-like projections that come off of these gustatory epithelial cells. And the taste will actually come in through, or the taste chemical will come in through the taste pore, bind to these gustatory hairs, and activate the gustatory epithelial cells. The other, it is important to note that the gustatory epithelial cells themselves, even though they have the receptor, are not a sensory neuron in itself. The sensory neuron comes very close to these gustatory cells, and what will happen is when these cells become activated, they will release a neurotransmitter onto the sensory neuron, and then the sensory neuron will become activated and carry that information to the central nervous system. So this shows an example where the sensory receptor is on a separate cell that will communicate with the sensory neuron. Over here, we're going to have the basal cells. These are going to be a set of supporting cells that will kind of help the gustatory cells. So those are the taste buds. Here's a little bit of a bigger picture so you can see some of the structures. So here in like this dark blue and light blue, we have the gustatory epithelial cells here that are going to have these gustatory hairs, these little extensions that come off of these cells. And this is where the chemical or chemoreceptors are going to be located. Again, and then in yellow over here, we have the sensory neuron, and the gustatory cells will send neurotransmitters to the sensory neuron to activate them when a particular chemical has bound to the chemoreceptors. And over here, we have the basal epithelial cells. This image isn't in your book, but I did want to give you another image that I think is really interesting and kind of beautiful. So over here, what we have done is we've actually taken a scanning electron microscope image of the tongue, and you can see the different papillae located there. And here is a light microscope image of the papillae. So we have, we have taken a histological cross-section of the tongue, and we're seeing the stratified squamous epithelium located here. And over here, we're starting to see the taste buds on the papillae. So taste can actually be described to have 5 different sensations, and these sensations are derived from what type of chemicals actually bind to the chemoreceptor. There will be a separate chemoreceptor for each of these sensations. And there will be a separate a taste bud for each of these sensations. So sweet is going to actually detect glucose. Glucose is a type of sugar, which is really important. It's going to be the main sugar that we use for creating energy and ATP in our mitochondria, and it's going to be our blood sugar. So sweet will detect glucose, so we can detect that we're getting enough of that blood sugar in our body. Other chemicals that mimic glucose will also taste sweet. This is what happens with sugar substitutes like Splenda and stevia. They have a similar but a structure to glucose, so they will also be able to bind to these sweet chemoreceptors. Sour, on the other hand, will actually bind to hydrogen ions, and because sour is associated with high acidity, and so things such as like lemons and grapefruit and limes are going to have a lot of hydrogen ions in them because they're acidic, and our brain will understand that and perceive that as sour. Salty is going to bind to sodium ions. So, and then bitter is going to bind to alkaloids. Alkaloids are going to be basic compounds that have a nitrogen group to them. And so these chemoreceptors are specially structured to bind to these basic compounds. And bitter is really important because many bitter things are poisonous or toxic. And so this is one of the reasons why we evolved a bitter sensation, is to detect these toxic compounds. But things such as coffee and cocoa beans and stuff like that can also be bitter, and so we've grown to enjoy some bitter compounds. And the last one is umami. Umami is a newer discovery. Umami actually is going to taste for things that are meaty or savory. And umami actually identifies amino acids, specifically glutamate in food. And it was discovered in the 80s, and umami translates from Japanese to English as delicious. So this is going to be found in things such as meat and tomatoes and Parmesan cheese. So things that have that very savory kind of flavor is going to have that umami sensation, because we're going to be detecting those amino acids in that food. So let's go into kind of the physiology of taste. I did write it out step by step for you, located here, but I do want to go through it on a diagram. So if you want to pause and kind of write this down, I'll give you a moment. Okay. And let's kind of talk about this together. So for your exam, these are going to be kind of essay options. So you can choose different sensations and describe the step-by-step physiology of that special sense. So how does the physiology of taste or gustation work? What will happen is food that we eat is going to dissolve in our saliva, and those compounds are going to coat our tongue, cheeks, esophagus, palate. And what will happen is that saliva will actually carry those tastants or those food chemical compounds through the taste pore. And what will happen is it'll bind to the gustatory hairs, and it'll bind to chemoreceptors that are specific to that particular compound. So if it is a sweet taste, it will bind to chemoreceptors that are designed to bind to glucose receptors. What will happen is if the correct compound binds to the chemoreceptor, the gustatory epithelial cells will become activated, and they will produce a neurotransmitter, and they'll release that neurotransmitter to the taste fibers, or these are the sensory neurons. And these sensory neurons will become activated and create an action potential, and that action potential will be sent to the central nervous system. So those are going to be the different steps of gustation. How does this information actually get to the central nervous system? And what kind of neurons or nerves are we going to use? So here we're really going to look at the taste or gustatory pathway. And in this pathway, we're going to focus on 3 nerves. So I divided it into kind of 3 parts or 3 slides. So first and foremost, what's going to happen is information is going to be carried away from the tongue, the pharynx, epiglottis, those regions of the mouth, through, and then it'll be carried to the medulla oblongata. To be able to carry it from the tongue to the medulla oblongata, we're going to rely on 3 different possible cranial nerves. There's going to be the facial nerve over here that will detect things on the front of the tongue. Things that are more posterior are going to be detected by the glossal pharyngeal nerve and be sent to the medulla oblongata. And the vagus nerve will detect things or detect taste in regions such as the pharynx or epiglottis that are not associated with the tongue. So the sensory neurons are going to be located in either the facial nerve, the glossopharyngeal nerve, or the vagus nerve. And all of these will be carried to the medulla oblongata. It will be carried to a specific location in the medulla oblongata that is associated with taste, called a solitary nucleus. So it'll go there. And it will synapse with the second neuron. The second neuron is going to carry information from the medulla oblongata up to the thalamus. Remember, most sensations, with the exception of olfaction, will actually travel through the thalamus and to be processed and organized before it can go on to the cerebral cortex. So here, the second neuron is going from the solitary nucleus of the medulla oblongata to the thalamus to get processed. And the last neuron, the third neuron, will synapse with the second neuron within the thalamus, and it will actually send that information from the thalamus to the gustatory cortex that is in the insular lobe of the brain. And there it will be understood and processed, and it can lead to conscious understanding of the sensation that we are experiencing. So that is the pathway for the gustatory sense or taste. So in our next video, we're going to focus on smell, but I hope this was helpful. If you have any questions or concerns, don't be shy to email me at my Keuka College email. And I wish you a wonderful day, okay? Be good to yourselves. Bye-bye.